Assalamu alaikum. Now we are going to discuss some guidelines for experimental research, part 2. The ILOs of this lesson. By the end of this lecture, we should be able to: number 1, recognize why we use animals in experimental research. Number 2, to realize the aim of preclinical animal studies in clinical development of drugs. Number 3, to establish the 3 R's rule in animal research. Number 4, to suggest what authors should clearly state in their experimental research. Number 5, to differentiate between in vivo, in vitro, and ex vivo studies. And the last point, to identify the ethical considerations in experimental research. Why we use animals in experimental research? Animals are used either to understand the pathophysiology of certain diseases. Of course, we first investigate the physiology, structure of the various systems and cells, and then the pathophysiology of certain diseases. They also used as a model. The animals used as a model for studying human diseases and to develop new treatments for these diseases. يبقى we either use animals to understand the disease, the pathophysiology, or to develop new treatments. Animals are biologically very similar to human. Mice share more than 98% DNA of the human. That's why we use mice and rats. Animals are susceptible to many of the same health problems, the same diseases like human. For example, cancer, diabetes, hypertension, heart diseases, depression. We can induce all these models in rats and study the effect of drugs and pathophysiology of these diseases. Also, rats and mice have shorter life cycle than human. And this is very important. The life cycle of the rats about 2 to 3 years, which equal 60 to 90 years in human. بمعنى ان تقريبا الاسبوعين في الرات في اللايف بتاعت الرات equal لسنة من الهيومن. if we wanted to study effect of drug or effect of chronic treatment, for example, for 2 weeks, this equal 1 year in human for average. So we can study throughout their whole lifespan diseases and across several generations, so as to understand the disease and to study the effect of drugs on these all ages. Animals should be used only if the scientific objectives of the experiments are valid. Valid means what here? That the experimental objectives have a reasonable contribution to human health, either by improving the understanding and the pathophysiology of the disease, or to improve the management or treatment of these diseases. And also if there is no other alternatives than animals, or to lower the cost. This is the clinical development of any medicine. According to the WHO, we know that there should be a preclinical animal experiments so as to test the acute toxicity, organ damage, dose dependence, metabolism, kinetic, dynamic, carcinogenicity. All these studies are called preclinical animal studies and very essential in development of any drugs. Before applying to patients or volunteers in phase 1. This is the preclinical. In experimental research, authors should clearly stated the following. Number one, the objectives of the research hypothesis and research questions, or the objectives. The reason of choosing this animal model and the species, strain, source, and type of animal. And should also state the details of each experiment being reported in this research, the study design and the number of animals, which is called the sample size, and the statistical methods used for analysis of your results. Animal research should consider 3 R's: replaced, refined, reduced. First, replaced. Animals should be replaced by less sentient alternatives such as invertebrates or in vitro methods whenever possible. What is the meaning of in vitro studies? And what is the difference between in vitro and in vivo? In vitro means within the glass. The experiments here are done outside the living cells in an artificial conditions that will be provided by the researcher, while in vivo means within the living organisms or cells and occur under precise cellular conditions prepared by the researcher. For example, in vitro, such as cell culture in petri dish or experiment in test tubes. In vivo means using model organisms such as mice, rats, guinea pigs, rabbits, cat, dog, and so on. Of course, in vivo is more expensive and more time consuming and needs more restrictions than in vitro. But in vivo is more precise and more accurate than in vitro experiments, because in vivo more resembling or modeling to the real life or the human. This is the in vivo and in vitro. In addition, what is ex vivo? Ex vivo studies means out of the living or outside the living organism. And this refers to an experiment or measurements which are done in or on tissue from an organism in an external environment with minimal alteration of natural conditions. For example, isolated organ path. We may do an isolated aortic ring. We take an aortic ring from the rat and do the experiment in the organ path, like the effect of acetylcholine, the effect of phenylephrine on this aortic ring to see the contraction and the relaxation and so on. This is the isolated organ path experiment. This is an ex vivo. Also, when we do an experiment and take tissue samples and then study histochemical or biochemical measurements in this tissue, this is an ex vivo, because we take the tissue from living organism and do an experiment or do measurements outside the living organism. So this is the second is refined. The first is replaced. The second R is refined. Refined means the protocol of the experiment should be refined to minimize any adverse effect for each of the individual animals. So we must use proper anesthesia in surgical intervention. The staff should be well trained. Housing should be suitable and high standard. And animals should be protected from any pathogens. This is the ethics for using animals. Reduce. This is the third R, reduced. The number of the animals should be reduced to the minimum so as to achieve the scientific object of the study. Too much animals or number will be loss of animals, and too little animals will lose data. So we should calculate sample size. And we will discuss how to calculate sample size in a separate lecture, inshallah. Ethical considerations: the use of animals in scientific experiments likely to cause pain, stress, or lasting harm generates an important ethical issue. There is an ethical considerations that should be taken and should be mentioned in the protocol. The plausibility of the study of the objectives. Choice of animal model is based on evidence. Housing conditions should be monitored and maintained. Sample size is kept at the minimum. Careful handling to minimize the harm or the pain. Also sacrificing animals under appropriate anesthesia, and samples should be carefully analyzed so as to maximize the benefit and never to lose data. Good luck and thank you.